Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. And guys, yes, we are back in Re-NEET Bounce Back series. But very important point guys, we have completed our class 11 biology, both zoology and botany. And you must know that every Wednesday and Sunday we are conducting Re-NEET exam. So I hope you are taking these tests every Sunday and Wednesday. Good evening all of you. So the link is already given guys. Quickly let me know. Are you ready for the class 12th syllabus now? Good evening. Yes guys. Just a second. Audible? Good evening, good evening. Guys, am I audible? Good evening, good evening. Guys, am I audible? Yes. What are the chapters that you're covering today? So guys, we are starting with class 12 syllabus today, and in 3 parts we are going to cover up our complete zoology. Now let me tell you which are the chapters and how we are going to cover up and what is going to be the scheme. So yes, you must know what we are going to discuss today. So in guys zoology, we are going to cover chapter human reproduction. This is what I'm going to discuss today, and the related chapter, that is reproductive health. And reproductive health. These are the 2 chapters that I'm going to cover today. So class will only be till 7 o'clock because immediately after this class I have a plus batch also, so won't be able to take a longer class. But tomorrow guys, tomorrow we are going to have a longer class in which we are going to cover 2 very big chapters. One is evolution and second would be health and diseases. Right? And then in the next class, I'm going to cover up microbes in human welfare. And then biotechnology 1, biotech 1 and biotech 2. So we are going to cover up these chapters in this format. So this is part 1. This will be covered, that is today. These will be covered in part 2, that is tomorrow. And then these 3 chapters will be covered up in part 3. So today is 26th. Yes. So today is 26th guys. 26th, 4 p.m. we are discussing this. Tomorrow, 27th at 5 p.m. we are discussing these 2 chapters. And 28th, 5 p.m. onwards, we are discussing these chapters. So this is going to be a schedule for completing class 12 zoology. Is that clear? Is that clear? Microbes today, not today. No, we are going to start with human reproduction and reproductive health. So reproductive health and human reproduction have to be discussed simultaneously. I could have taken microbes today, but as I've already told you, this class wasn't really planned, right? So I have a class, I have a plus class after this. So there'll be some time constraint. So guys, let's quickly jump on to our topic. Today's topic, that is human reproduction. We are going to discuss everything in detail and from 0 to hero level, right? So please let me know that you're ready with your NCERTs and your notes, and we can start with the chapter. Quickly let me know. So guys, I am exactly using the same content which we discuss when we discuss the topic from the 0 level, right? So we are discussing everything in detail in these sessions. So don't forget to like, subscribe, share all these sessions with your friends. When will you take the next special class? I will, husband. I'll align the class soon. Is that clear? Are we ready? All right. So guys, starting with human reproduction, what is human reproduction? We know it is a type of sexual reproduction. So humans are sexually reproducing organisms. Talking more about human reproduction, so we are unisexual. We are sexually reproducing organisms. Sexually reproducing organisms. And in sexually reproducing organisms, guys, and we show direct development, that means no larval stage. In our case, there is internal fertilization. So these are some characteristics of human reproduction that you must know. Is that clear? Now, talking about the events of human reproduction. Always and always, guys, whenever there is sexual reproduction, so in sexual reproduction, guys, these are the steps or the events that would always occur, be it plants or it animals. All those organisms who undergo sexual reproduction will show these few events. Number one, gamete formation, which is called gametogenesis. Then transfer of gametes. Now this transfer of gametes occur during the act of mating or copulation, right? Then we have the third step. Third step is fertilization, which is also known as, guys, syngamy, fusion of gametes, right? And in our case, the syngamy is internal. It occurs within the female body, hence it is internal, followed by now fertilization will give rise to zygote formation, right? And this zygote will be followed by embryonic development, and then once the embryonic development is done, there will be childbirth or parturition. Please tell me all these points are clear. Yes, is this point clear? All right. Now guys, out of these five steps, please listen. Out of these 5 steps, the first 2 steps, these constitute pre-fertilization events. These are the pre-fertilization events, right? While these 2 steps constitute the post-fertilization events. Is it clear? So these constitute the post-fertilization events. Please tell me all these points are clear. Shall we move on now? Moving on now. All right. So guys, since we are unisexual, so both male and female structures will be present in different bodies. Sexes are separate. So before going on to the events, we have to understand the architecture. We have to understand the structures of structures which are involved in human reproduction. So we'll take male reproductive system and female reproductive system separately. Now first of all, guys, male reproductive system is located in the pelvis region. Pelvis means hip region. This is found in the hip region, right? Sorry, found in the hip region. Are we clear? Then what, not only within the pelvic region, but there will be some structures which are intra-abdominal. That means they are found within the abdomen, and there will be some structures which are extra-abdominal. We'll talk about them one by one. Now, what are the components of the male reproductive system, guys? So, male reproductive system consists of a pair of testes, accessory ducts, accessory glands, and external genitalia. Right? Now, out of this, the pair of testes, this constitutes the primary sex organ, also known as gonad, because this is the place where actually sperm production, that means the gamete formation, is going to occur, while all the other structures, ducts, glands, and external genitalia, they will constitute the secondary sex organs. Are we clear? Are we clear till here? Yes. So pair of testes, ducts, glands, and genitalia, these constitute the secondary sex organs. And now we are going to discuss each structure one by one. Please tell me these points are clear. Now starting with testes, also known as testicles, they are found in a pair. They are soft, they are smooth, they are oval, they are pinkish, and they are male primary sex organs, just told you. Now, what are the functions of testes, guys? What are the functions of testes? There are two functions, two main functions. Number one is sperm production, which is called spermatogenesis. Is that clear? And second is hormone formation, or hormone production. So yes, it does act like an endocrine tissue also. It is the endocrine function. Now the main hormone produced by testes is testosterone. This is the main androgen. This is the main androgen, male hormone. Are we clear? Please tell me. And it is this testosterone which is responsible for, of course, sperm production as well as masculine characteristics. Are we clear? Please tell me, are we clear here? Right? Now guys, the point is, where are testes located? So remember, testes are located outside the abdominal cavity. Where are they present? Outside the abdominal cavity, in a special pouch known as scrotum. In a pouch known as scrotum. Why? So that means testes are not present within the abdominal cavity. Hence, can we say, since they are present outside, we can say testes are extra-abdominal. Testes are extra-abdominal. And where exactly? They are present in the scrotum, a pouch-like structure. Were they always outside? No. So testes were not always present outside the abdomen. Earlier, till 7th month of pregnancy or gestation, testes are intra-abdominal. Testes are intra-abdominal. It is later on, during the after the 7th month, that they descend into the scrotum. Is that clear? So later on, they descend into the scrotum. Now, what is the purpose? The reason? The reason is, guys, sperm production requires lower temperatures. It provides lower temperature. How much lower temperature? 2.5 degrees Celsius lower than the normal body temperature. Because sperm production cannot occur at 37 degrees Celsius. Now remember, see, sperm production requires a lower temperature, but sperms can be stored. Sperm production at lower temperature, but sperm storage can take place at normal body temperature, that is at 37 degrees Celsius. So they are produced at the lower temperature but stored at a normal body temperature. Please tell me these points are clear. Are we sorted? Are we clear till here? Anyone with any doubt? Yes. So you can see it in this diagram, guys. So these are these structures. This is testes. And do you see this external pouch? This external pouch represents the scrotum. This represents the scrotum. Are we clear? Yes or no? Are we clear till here? Right? Now, so testes, guys, they are connected to the abdominal cavity through inguinal canal. Now, when we say that testes were earlier intra-abdominal and then gradually descend into the scrotum, then there must be some passage which is connecting the abdominal cavity to the scrotum. Now, what is that? See here. So, the cavity of scrotal sac is connected to the abdominal cavity through inguinal canal. It is connected through the inguinal canal. Is it clear? Let's see it here. So this is the inguinal canal, this area. This is how the testes are going to, this is the canal through which testes are going to descend into the scrotal sac. These are the testes. Are we clear? Please tell me. Now, in case this does not happen, failure of descending, if testes fail to descend in the scrotum, into the scrotum, then what will happen? Temperature regulation will not take place, yes or no? Temperature regulation will not take place, sperm production will not occur, right? And this can lead to infertile sterility, and the condition is known as cryptochidism or undescended testes. So what happens? So the male child is born infertile. This is infantile. This is going to cause infantile sterility. But these days, this is not a big issue. Why? Because even if this cryptochidism is detected, then through inguinal canal, which might be blocked. Is that clear? It is connected. If testes fail to descend, that can be due to inguinal hernia or any blockage, inguinal hernia. Now, the testes fail to descend. But then what happens? These days it is not a big issue. We can surgically open up the inguinal canal and descend the testes into the scrotum. But this can be #corrected surgically. Right after the birth, corrected surgically. Please tell me these points are clear. Please tell me these points are clear. Yes, guys. So what is the purpose of scrotum, guys? What is the purpose of scrotum? The purpose of scrotum is provides lower temperature. That means thermoregulation. So what is it doing? It is regulating the temperature for sperm production. So the main function is thermoregulation. Please tell me these points are clear. Everybody, please tell me these points are clear. Are we sorted? Can we move ahead? All right, guys. So this is cryptochidism, which can be connected. Now see, let's see here again. So this is the scrotum. This whole sac-like structure is known as scrotum, right? And within the scrotum, guys, these structures are known as testes or testicles. These are testicles, right? So testes are extra-abdominal. Now there are many ducts that arise from the testes, which are going to carry the sperms back into the abdomen for storage and for removal from the body as well. Now there are certain other structures also. See here, there are certain other structures like these structures, right? And then along with many other structures, they are going to form a spermatocord. So basically, through inguinal canal, lots of blood vessels, nerves, lymph vessels, right, and many accessory ducts also pass through. Now together, guys, they constitute the spermatic cord. So what is a spermatic cord? Spermatic cord consists of, guys, arteries, veins, nerves, vas deferens, and the connective tissue. Is that clear? So these are the components of spermatic cord. And where does it pass through? It passes through inguinal canal. Passes through inguinal canal. Please tell me these points are clear. Are we clear till here? So it runs from the abdomen down to each testicles through inguinal canal. Yes. So these are the components, as we can see here. So this is all spermatic cord. This is all spermatic cord. And what does it consist of? It consists of arteries, veins, nerves, vas deferens, right, and other tissues, and also the connective tissue. Please tell me these points are clear to everyone. Yes? Can we move on now? Can we move on to the next topic? Is that all clear? Perfect. All right, guys. Now, moving on to human reproduction, the internal structure of testes and the function I've already told you. These are the functions. Function number 1 is spermatogenesis, and second is the endocrine function, that is production of testosterone. Is that clear? Now, these are the testes, as we can see in the diagram. These are the testes, and this is the spermatic cord basically, which is connecting abdomen to the scrotum. So, and here lies the scrotal sac. This is the scrotal sac, and the passage through which it is passing back into, so this passage guys is the inguinal canal. Are we sorted? Are we clear here? Now moving on to the internal structure. The functions are clear, the location is clear to everyone, and now coming on to the internal structure of testes. So let's have a look. So guys, they are oval structures, pinkish oval soft structures, right? Now each testes, look very carefully, each testis consists of guys these structures which are called testicular lobules. These structures known as testicular lobules. So these are testicular lobules. Now as a matter of fact, each testis has how many lobules? Around 250 in number in each testis. In total 500. So this is the testicular wall. This is the testicular wall, and within the testes we have testicular lobules. How many are they? They are 250 in number. Are we clear? Now within these testicular lobules guys, we have a very important structure. We have these seminiferous tubules. We have these seminiferous tubules. And how many of these seminiferous tubules? There will be 2 to 3 seminiferous tubules in each. Is that clear? I'm just drawing one. There will be 2 to 3. So these are seminiferous tubules, and seminiferous tubules are the, these tubules are the unit of testes. We call them the unit of testes. Why unit? Because this is the location where actually sperm production takes place. And how many? 1 to 3 in number in each lobule. So that means in total one testes will have roughly 250 to 750 seminiferous tubules. Yes or no? Right? So guys, now ahead of this we have these straight ducts. So starting from the seminiferous tubules, we have these straight ducts. These are all straight ducts. Is that clear? Now these straight ducts will all open up in this. These are straight ducts. I'll write down straight ducts. Now all these straight ducts are going to open up in this network and form this network of ducts known as rete testes. Known as rete testes. Are we clear? Are you able to see the passage? And from rete testes guys will arise 12 to 15 small ducts, and these ducts are known as vasa efferentia. Is that clear? So seminiferous tubules are going to produce sperms. From straight ducts they move into the rete testes, and from rete testes sperms go into the vasa efferentia, and vasa efferentia is going to carry sperms outside the testes for the very first time. Please tell me these points are clear. So vasa efferentia is going to carry the sperms outside the testes for the very first time, and now all these are going to open. Wait, I'll draw it with a different color. So all of these are going to open up in a highly coiled structure guys, and this highly coiled structure, this is highly coiled structure around 1 to 2 meter long, super coiled. This structure is known as epididymis. This is known as epididymis. Are we clear? And from epididymis guys arise that tube which will carry the sperms back into the abdomen for storage. This is where the sperms will be stored. Are we clear? Please tell me. Now all this structure, this whole structure is present in the scrotum. So all the structures I have drawn here, they will be present within the scrotum. So testes along with the ducts, then this vasa efferentia, then epididymis, they will be all present in the scrotum. And from here arises the vas deferens. A part of vas deferens is also extra abdominal that is present in the scrotum. Please tell me these points are clear to everyone. Is that clear? Right. Perfect. Now, moving on. So this is the internal structure, guys. Now, like every other vital organ, there must be a protective covering here also, right? So there are 3 different types of protective coverings, guys. But, so this is vaginalis, tunica vaginalis. But the main covering of testes, which NCERT has mentioned about, is this, which is present just below the testicular wall and surrounds the testes from all the directions. Now this is known as tunica albuginea. So this is the protective covering. Although there are 2 other coverings also, but we have to talk about NCERT and the main covering of testes, that is tunica albuginea, the protective covering. Is it clear? Where is it present? It is present just below the testicular wall. Now we will see the same covering in case of ovary as well. So present just below the testicular wall, and we will be able to see exact same covering in ovaries as well. Is that clear? Please tell me. Thank you, Vrinda. Yes, guys, are we clear? Yes or no? Now, moving on. So I have told you the structure, right? So this is a diagram, a separate diagram that I've used. Now see here. So yeah, so these are the see, this is the main protective covering, that is tunica albuginea. There is outer tunica then there is tunica vaginalis, and then tunica vasculosa also, but we can ignore this. Then guys, we have these testicular lobules. These ones are the testicular lobules, right? These are the testicular lobules. Sorted? And do we see highly folded structures called seminiferous tubules? And ahead of seminiferous tubules, guys, we have straight ducts. These are the straight ducts, and from straight ducts it opens into this this network known as the rete testes. And from rete testes, do you see these tubules arising? We call them afferent ductules or vasa efferentia. Yes, and vasa efferentia is going to open up into this highly coiled structure. Do you see how coiled, highly coiled it is? This is all epididymis. We don't have to learn the sub parts of epididymis, right? And then ahead of that, we have ductus deferens, simply known as vas deferens. Please tell me these points are clear and the diagram is sorted, right? So guys, these were the structures present in the scrotum. Now see, each testis is enclosed in a fibrous envelope, told you, that is tunica albuginea, which is fibrous and the protective covering, and it extends inward to form the septa and divides the testes into testicular lobules. So this is basically tunica albuginea that forms the lobules. Do you see these yellow portions? So this is all tunica albuginea going into it and forming the lobules. Are we clear till here? Yes or no? Yes? Now guys, now within these seminiferous tubules, let's see. Now within these seminiferous tubules, guys, if I look at a seminiferous tubule, we know every lobule has 1, 2, 3 seminiferous tubule. When we say it is a tubule, so it means it is going to be hollow from inside. The whole hollow portion is known as lumen. So there will be these structures, lumen, right? So another lumen. So I have drawn basically seminiferous tubules here. These are the seminiferous tubules. Are we clear? These are the seminiferous tubules. That's how they will look like. Seminiferous. Clear? Now outside the seminiferous tubule, guys, this is all interstitial fluid, or you can say interstitial space. This is all interstitial space. And in this interstitial space, guys, we have some special cells known by the same name, that is interstitial cells. Because they are present in the interstitial space, we will call them interstitial cells. And it was Leydig, the name of the scientist who discovered them, who studied them. We call them Leydig cells also. Now, what is the main function of interstitial or Leydig cells, guys? So, function of the Leydig cells is that these cells are going to produce testosterone. They are going to produce testosterone or the male androgen. Please tell me these points are clear. Yes, please. So, actually, testosterone is produced outside the seminiferous tubule. Yes. So, these are the interstitial cells present in the interstitial space producing the testosterone. Are we clear? Now, this testosterone will go inside the seminiferous tubules and help in sperm production. Now, see, this is the seminiferous tubule. Is that clear? Now, if we go into the details of seminiferous tubule, see, each seminiferous tubule is lined by germinal epithelium. So, this is all lined by germinal epithelium. So, this layer that you see in here, this whole layer, this outermost layer, this whole layer is known as germinal epithelium. Please tell me these points are clear. Is that clear? All these are germinal epithelial cells. Are we clear? Now, see guys, germinal epithelial cell consists of two types of cells here. So, germinal epithelium consists of two types of cells. One is the Sertoli cells, again in the name of scientists, Sertoli cells, and second is the spermatogonia cells, also known as sperm mother cells, and we obviously understand the function of spermatogonia here, right? Now, both these cells are diploid, of course. Now, as the name itself indicates, sperm mother cells are going to produce sperms by the process of spermatogenesis, but it is not a single step process and we are going to discuss the details. Clear? And then Sertoli cells, also known as nurse cells, because they nourish. They are also known as sustentacular cells. Sustentacular cells. So, as the name says, nurse, so they are going to nourish. This is the first and the foremost function of Sertoli cells. Are we clear till here? Please let me know. Are we clear, guys? Are we clear? Nutritional, yes, absolutely. But there are some other functions also performed by the Sertoli cells. Now, see, Sertoli cells are more of glandular cells. These are glandular as well as, these are columnar cells. These are columnar cells, while spermatogonia cells are cuboidal cells. So, cuboidal epithelium, while this is the columnar epithelium. So, see guys, structural organization is more, the study of structural organization is beyond that one chapter of animal tissues. You have to know where each and every type of cell is present. Are we clear? Are we clear? Please let me know. Is that clear, guys? Please tell me. Are we sorted? Right? All right, guys. Now, let's see how they are present. I'm going to show you here in this diagram and then going to go into this detail. So, these are the Sertoli cells, guys. Do you see? These are the Sertoli cells. And rest of these are the spermatogonia cells. These are the spermatogonia cells. Yes. So, gradually spermatogonia cells are going to form sperms and there will be intermediate stages and that have been shown here, but we will study them in more detail. Please tell me these points are clear to all. Please tell me. Yes, guys. So, germinal epithelium consists of undifferentiated male germ cells called the spermatogonial mother cells, and then Sertoli cells, which are meant for nourishment, Other than nourishment, guys, Sertoli cells are also going to concentrate testosterone. So, and it is also going to produce inhibin. It is going to produce androgen binding protein that concentrates testosterone within the seminiferous tubule. Yes, right. It is also going to help in meiosis 2 of oocyte, right? So anaphase promoting complex also comes from here. So there are multiple functions, but the main function is nourishment of the developing spermatocytes. Are we clear, guys? Right? Now, interstitial spaces, I've already explained. There are regions known as interstitial spaces, and in these spaces, guys, we have interstitial or Leydig cells. And what do they do? These interstitial spaces have interstitial cells, also known as Leydig cells, and we've already studied what they do. They produce testosterone. So testosterone is produced by the outer cells but used by the internal cells for spermatogenesis. Is that clear? And what is the function? We have already read male secondary sexual characters as well as the spermatogenesis. These are the two functions of testosterone. And spermatogenesis is also is always a primary sexual character. Is that clear? It is a primary sexual character. While what are these secondary sexual characters? Secondary sexual characters means facial hair growth, pubic hair, right? Then hoarse voice changes, vocal cords become thicker, right? Then muscular and skeletal development, muscular development, then broad skeleton, shoulders particularly, right? Then action on your oil glands. So there are so many functions performed by them. Development of the genitalia, right? These are some of the secondary sexual characters. Please tell me these points are clear to you all. Is this point clear? Monica, yes, now you join the class, and we have just started with this topic of human reproduction, and we are still studying male reproductive system. Clear, guys? Now coming on to epididymis. Told you this is vasa efferentia. Vasa efferentia leaves and forms this highly tightly coiled tube. Sorry, this forms tightly coiled tube, 1 to 2 meter in length. But what is the function, guys? Now, what is the function of epididymis? Now, see, we know sperm production occurs in seminiferous tubule. From here, they go into the straight ducts to vasa efferentia, vasa efferentia to vasa efferentia to epididymis. So, epididymis is that place where there is temporary storage of sperms, right, for around a week. And this is the place during this storage, sperms undergo maturation. It is here that they get motility and become motile. If this phase would not occur, then the sperms would never become motile. Please tell me these points are clear to you. And this happens in epididymis. See here, located in the posterior part of each testis, provides temporary storage, gives time to mature, and during maturation they acquire motility. Please tell me these points are clear to you all. So we don't really have to go into the details, but like the different parts of epididymis not required, but the last part of epididymis is going to change into the vas deferens. So it passes through vasa deferens, it passes through the inguinal canal into the abdomen and loops over the urinary bladder. So vas deferens, so guys see here, so this is the vas deferens. Do we see here? Do we see this duct? So this is the vas deferens, and this vas deferens, see here, this is the vas deferens. Vas deferens is going to carry the sperms back into the abdomen. Okay, so which duct carries the sperms back into the abdomen for storage? It is vas deferens. Clear? I told you in the very beginning, the sperm production requires lower temperature, but storage can occur at the same normal body temperature, that is 37 degrees Celsius. So vas deferens carries the sperms back into the abdomen, back into abdominal cavity or abdomen. That is one. This is the place where sperms will be stored permanently before they are ejaculated. Is it clear? So this is the site of permanent storage. So what does permanent means? Till ejaculation. Permanent storage of sperms till ejaculation. Is that clear? Permanent storage. Please tell me these points are clear to all. Yes, yes, yes. Please tell me, guys, these points are clear to everyone. Yeah? All right. Now, so see here, once again, I want your attention to this diagram. Do you see that this part of vas deferens is getting swollen? It dilates. This dilated part is known as ampulla. This is the ampulla of vas deferens. This is where the sperms are actually stored. This is where the storage occurs, in the ampulla. Yes or no? So this was about vas deferens, guys. And then also, now next thing, what happens here? So it's showing you the structure. Now see, we have just discussed that this is the ampulla of vas deferens. Yes, this is the ampulla of vas deferens. Now do you see that there is a small duct arising here? Can you see this duct? Can you see this duct, guys? So this duct, this green colored duct that I have drawn here, this duct is called the seminal vesicle. Now it receives the duct of seminal vesicle, and together, guys, so there is this duct of seminal vesicle, and together they are going to form this, wait, there is this duct of seminal vesicle and this is vas deferens. So both of them, this is the vas deferens, and then there is this duct here, this is the duct of seminal vesicle. Together they are going to form this small duct known as ejaculatory duct. So how is ejaculatory duct formed, guys? It is formed by the union of vas deferens plus duct of seminal vesicle. Please tell me these points are clear. So it is formed by the union of vas deferens and duct of seminal vesicle. Is that clear? Also known as ductus ejaculatorius. So why do we call it ejaculatory duct? Because this duct is going to receive sperms only during ejaculation. And I've just told you, till ejaculation, sperms will be stored in the vas deferens ampulla region. So if sperms are present here in the duct, they are going to be ejaculated. Are we sorted here, guys? And then beyond this ejaculatory duct, listen here, beyond this ejaculatory duct, guys, we have ahead of this, we have this duct called, so this duct will be known as urethra. Now this duct will be known as urethra, right, which will open at the tip of the penis. Please tell me these points are clear, right? Now see, this is another great explanation of ejaculatory duct. Do you see the ductus deferens? This is the ampulla region, this is the ampulla of vas deferens, and this is the seminal vesicle. Do we see seminal vesicle? This is the duct of the seminal vesicle. right? Now together, both of them, this is your vas deferens. This is the vas deferens. So vas deferens and this together form this small duct, which is known as ejaculatory duct. So we have two ejaculatory ducts from each side, and they will open here in this region called urethra. Please tell me these points are clear. Please tell me these points are clear, guys. Right? So there are two ejaculatory ducts. In this, they converge at the central prostate gland. They carry sperms from vas deferens, and from the seminal vesicle it is going to have the fluid, and this will help in the transport of sperms to the outside. Please tell me these points are clear to everyone. Now guys, what is the passage? So we have read enough about where the sperms are produced and how they are going to travel through the testes and outside the ducts till they are released. So what is the passage of sperm? So as passage, it starts from the seminiferous tubule. Starts from the seminiferous tubule. We know there are straight ducts which will carry sperms outside the seminiferous tubules. And all these straight ducts will open into this structure called rete testes, and from rete testes arise vasa efferentia. Yes, from vasa efferentia, guys, begins the highly coiled structure called epididymis, and epididymis will form a dilated structure or a broader duct known as vas deferens. Yes, then vas deferens will join the duct of seminal vesicle, and together these will form the ejaculatory duct, and the two ejaculatory ducts will open into the urethra. Now see, in males this urethra is long and a common passage for sperms as well as urine. That is why in males we call urethra as urinogenital duct. Yes, and we have read the same terminology even in case of frog. So we see excretory system and reproductive system of males getting converged. So guys, now here, see, till vasa efferentia, so here, sorry, till vasa efferentia, the sperms were within the testes. So it was vasa efferentia which carried sperms outside the testes for the first time. So guys, this represents the intratesticular pathway, while from vasa efferentia onwards, this represents the extratesticular pathway. Please tell me these points are clear to everyone. Yes, please. Please tell me these points are clear to everyone. Right? Now, moving on to, see, I already told you this is the urinogenital duct, starts from here and then opens to the tip of the penis. Right? It is a shared terminal duct. Terminal means last duct between reproductory and urinary system, and that is why, guys, that is why we call it urinogenital duct. Clear? Starts from the base of the bladder to the tip of the penis. Clear to everyone? All right. Now, talking about the genital glands. See, we have already discussed about the primary sex organ here. So the primary sex organ is the testes, and we see all these structures lie within the testes. That is why we call it intratesticular pathway. Yes, but all these ducts, starting from straight ducts and vasa efferentia, guys, all these represent, see, all these, including these represent the accessory ducts. All these are the male accessory ducts. Yes or no? Except for this, which is a gland. So we'll be discussing about this in the male accessory glands. Now, talking about the glands, so we have three types of glands here. One is seminal vesicle, then we have prostate gland, and then we have bulbourethral gland, also known by the name of Copper's gland in the name of scientist. The first thing, which gland is paired and which is singular? So this is a single gland, so that means 1. These are paired, that means 2 in number. Is that clear? Now together their secretion is known as, see, these glands constitute the seminal plasma. So there should be no confusion about it, that NCERT has used the term seminal plasm for the secretion of all the 3 glands. Please be very careful. So these glands constitute the seminal plasma. So all 3 glands. Is that clear? According to NCERT, seminal plasma is the term for the secretion of all the 3 glands. And what is it rich in? It is rich in fructose, calcium and certain enzymes. They have not given a lot of details. Now see, what is the function of fructose going to be? This fructose is going to be energy source. Like our cells love glucose. So fructose is present in the seminal plasma and this becomes the energy source. So they are going to break down fructose to form energy. Also guys, you must know that fructose as an energy source or as a carbohydrate is not present in any part of the body other than seminal plasma. Is that clear? You don't have fructose in your brain fluid or anywhere else, not even in blood. Fructose is present only and only in the seminal plasma. Please tell me these points are clear to everyone. Is that clear? Now, moving on to the third, particularly bulbourethral glands, also known as the Copper's gland, it produces a secretion which helps in lubrication of penis. Is that clear? So see here, where are the structures present? So we have studied already, this is the vas deferens, right, going back into the abdomen like this. Remember, this is the vas deferens, this is a seminal vesicle, they'll be forming. And now, seminal vesicle is present here, remember? And it is this seminal vesicle whose duct fuses with vas deferens to form the ejaculatory duct. So one, and then there'll be another seminal vesicle. So these are 2 in number. Then we have this one common prostate gland. Is it clear? So prostate gland is the largest of them all and it is a chestnut shaped gland present around the urethra. So see, this is the urethra going on. This is the urethra, it is mentioned here. Around the urethra, just below the bladder. So where is this present prostate gland? Just below the bladder. Surrounding the urethra is this prostate gland. Now the urethra continues and enters the penile region, so that like this, and it will open at the tip of the penis, right? Now here guys, here let's draw it here. Here we have the third gland. Which is known as bulbourethral gland or Copper's gland. Please tell me these points are clear to everyone. Yes, please. If I start now for NEET from 229, how much can I reach maximum, man? Shalini, are you looking forward to 2027? Or you took the exam in 2026? Please tell me this. Snehita says, if you are consistent, you can score 300 to 320 above in biology. That is very true actually. Even if you are NEET 2026 student, you took this exam and somehow this whole thing has happened and you were a prey to that, right? And you took a lot of time coming back to the normal senses that, okay, now I don't have to give up and come back and make a comeback as well. You want to bounce back. So see, remember, even wherever you are, whatever marks you have scored, you are certainly not at 0. Yes, you're certainly not at 0. So that's what you have to remember, guys. This is bounce back. So you are at a certain level. This opportunity is going to help you get better marks. You getting it? So if you are already scoring around 200 plus, you can easily score around 300, 320, even in this 1 month. No, although not 1 month is left now, but still, you're not at zero. Just remember this. So you don't have to get disheartened and feel so low and as if you're just starting. No, you already have a certain level and you're already at a good level. And guys, now coming to the external genitalia of males, which is the penis. Penis represents the male copulatory organ and male external genitalia. Now, why do we call it external genitalia? Because it is present outside the body. So hence it is of course extra abdominal. This is also extra abdominal. What are the functions, guys? What are the functions? So it is a copulatory structure. This is a copulatory organ. This is function number 1. Extra abdominal. And so what are the external genitalia structures? One is penis and second is scrotum. Is it clear? So it is a copulatory organ and is going to help in the release of sperms into the female or outside. Now, so penis consists of one very important tissue, write down, which is known as erectile tissue. Consists of erectile tissue, and this erectile tissue is going to help in, as the name says, it causes erection. It causes erection. What does erection means? Hardening of the penis. Right? Now this is important for the insemination. This erection is important for insemination, that is deposition of semen in the female tract. Is it clear? Also, you must remember this erection is completely a vascular phenomena. So basically what happens during erection is there is rich blood supply to the structure. Vascular phenomena means rich blood supply to the structure. So rich blood supply causes the hardening of the structure, which is known as erection, and it is important for insemination, that is deposition of semen into the female tract. Now, erectile tissues are of two types, guys. They are of two types. So one, we have corpus cavernosa, and second, we have corpus spongiosum. Is it clear? So second is corpus spongiosum. Now see, if I show you through a diagram, listen, corpus cavernosa, yes, it is paired, it is upper and it is dorsal. Corpus spongiosum is one, single. It is ventral and it allows the passage of urethra. Is that clear? And also the tip, forms the tip of penis, which is called glans penis. Now see, if I go into the details here, so how does it look like? So basically, so this is the transverse section of penis, and we have these two structures present on the dorsal side, which are called corpus cavernosa. And then we have this structure called corpus spongiosum. Is that clear? These are 2 in number, these are 1, and they are all cylindrical rod-like structures. And through this cylindrical rod-like structure passes urethra. Is this clear? So urethra is surrounded by corpus spongiosum. Please tell me these points are clear. So there's a rich blood supply which causes the hardening and straightening of penis known as erection. Is that clear? So already discussed, it is external genitalia. What is the function? Helps in insemination, made up of special tissue, that is the erectile tissue, and which helps in the erection to facilitate insemination. Enlarged end of penis, the tip of the penis is called glans penis. And what is this penis made up of? Glans penis tip is made up of corpus spongiosum. Now glans penis is covered by a loose fold of skin. See, it is covered by a loose fold of skin known as foreskin, or we call it prepuce. We call it prepuce. So there is this loose fold of skin present here, and this is known as foreskin. Please tell me these points are clear to everyone. Yes guys, are we clear? Please tell me these points are clear to all. So guys, this was all about the male reproductive structure. Is that clear? The male reproductive system. All the structures have been studied, and now coming on to the functional part. Now we know there are two functions performed by testes. One is the production of testosterone, and second, we've seen the function, and second is the sperm production, that is called spermatogenesis. So let's talk more about spermatogenesis. How and when does it start? So first of all, where does it occur? Where? So where? Answer is in seminiferous tubule. In seminiferous tubules. Who produces it? That is the sperm mother cells, also known as spermatogonia. Is that clear? Now, then when? Answer is when does it occur? So the point is, it starts at puberty. It starts at puberty, and then puberty, onwards, it goes on lifetime. Goes on lifetime, although with age it reduces and the rate of production reduces. Is it clear? So where? Seminiferous tubule. Who produces sperms? That is spermatogonia. When? It starts at puberty. Why it starts at puberty? Why at puberty only? So why? Because of our biological clock. That means hypothalamus produces GnRH, gonadotropin releasing hormone. So this is also a part of biological clock, right, which stimulates the anterior pituitary to produce gonadotropins like FSH and LH. So basically, your hypothalamus and anterior pituitary are going to stimulate the production of sperms during this phase. Are we clear? Please tell me, are we clear here? Yes, everyone? So next question is how. So next question is how. So let's see how. So guys, first of all, guys, we have the sperm mother cell. Yes. So first of all, we have spermatogonia, or the sperm mother cell. Is that clear? This is the sperm mother cell, and we know it is 2n in number. So first of all, these sperm mother cells are going to undergo lots of mitosis to form more sperm mother cells, right? So they will keep on dividing and so on, right? Now see here. So they will keep on dividing like this. So do you see there are two types of cells formed? This is regularly undergoing lots of mitosis. And do we see there are two types of cells formed here? This is type A and this is type B. Now one of them continues to divide and the other stops dividing and enters the next phase, which is called the growth phase. Now see, from here, all these type B cells, these B cells, they will enter into the next phase, which is called the growth phase. All of them are going to enter the next phase, which is going to be the growth phase. Now NCERT has not really shown you like this type A and type B, but they have definitely mentioned it In the chapter of plant growth and hormones, if you remember, we have one exponential growth and logistic growth. Do you remember? It is clearly written there that when one of the daughter cells retain the ability to divide and the other goes on differentiating. Now see, this goes into maturation and gradually maturation or differentiation. Is that clear? So this phase, guys, this phase is known as, this whole phase, when it is just dividing mitotically, this phase is known as maturation, phase of multiplication or multiplicative phase. This is multiplicative phase. So what really happens during multiplicative phase? So sperm mother cell will form more sperm mother cells or spermatogonia. These are all sperm mother cells, right? So obviously the ploidy of each sperm mother cell is going to be 2n, and this goes on like that, right? Now these type B cells, so basically all these sperm mother cells are going to enter into the next phase, which is called the growth phase. So what really happens during the growth phase? So each cell is going to get bigger. It is going to get bigger like this. Is that clear? So I'm just going to continue with one of the cells so that you see what happens to each and every cell. So I hope you will be able to understand that this each is going to have the same fate. So let's say the sperm mother cell enters the growth phase, grows bigger in size, and that is when we call it, guys, instead of spermatogonia, now we call it primary spermatocyte. Can anyone tell me in the chat what will be the ploidy of a primary spermatocyte? After it has grown bigger, what is going to be the ploidy here, guys? 2n, yes. So this is also going to be 2n because it has only grown in size, right? And after growth phase, guys, it enters the next phase, and this next phase is known as maturation phase. Now during the maturation phase, it's going to undergo meiosis. What type of division occurred during multiplicative phase? It was mitosis. What happened during growth phase? Just the growth, right? But in maturation phase, it is going to undergo meiosis. So meiosis, as we know, occurs in two steps, meiosis 1 and then meiosis 2. Now see, when meiosis 1 occurs here, it gives rise to two cells, and each cell is known as secondary spermatocyte. Each cell is known as secondary spermatocyte. Now let's have a recapitulate. And please let me know, guys, please let me know what will be the ploidy of secondary spermatocytes now. What will be the ploidy of secondary spermatocytes? Since meiosis 1 has already occurred and this is the reductional type of division, yes. So what will be the ploidy here? It will be n. Each secondary spermatocyte will be n, yes. Now each of them will further undergo meiosis 2. Each one of them will undergo meiosis 2. Yes, meiosis 2, meiosis 2. And after undergoing meiosis 2, guys, they are going to form these structures known as spermatids. And once the number has already been reduced, it will be maintained. This will be the ploidy. Now, can you solve some numericals here? Can you see the ratio in which the cells are increasing? So you have one sperm mother cell, it will give you one primary spermatocyte, but that will give you two secondary spermatocytes and then 4 spermatids. Do you see this ratio here? Yes. Do you see this ratio, everyone? So we have one sperm mother cell giving one primary spermatocyte, giving us 2 secondary spermatocytes and giving us 4 spermatids. Yes or no? Do you see this ratio, everyone? So 1 gives us 1 and this 2, and these will be 4 in number. So can we remember this for the numerical purpose, guys? Please tell me. This is for numericals. Can you use this ratio? Please tell me. Right? Now, also, please see. Now, once the spermatids are produced, the sperms have not been produced yet, guys. These are spermatids. Now, the spermatids must undergo one more change. So, these spermatids, each one of them, they are going to undergo differentiation. They are going to undergo more changes in their structure and form a typical sperm-like structure. Yes, now these changes are known as spermiogenesis. This last step is called spermiogenesis. What is spermiogenesis? Where spermatids change into sperms. Metamorphosis of spermatids into sperms is called spermiogenesis. Please tell me these points are clear. So guys, this in total is called spermatogenesis. Please tell me if all these points are clear to everyone. Yes, guys? Are we clear? Right? All right. Now, as I said, this whole sperm production is controlled by hormones. So, if we talk about not only every monthly time, but if we start with how it started for the first time, like your hypothalamus, it produces a hormone, the releasing hormone known as gonadotropin releasing hormone. Yes, from this comes, from hypothalamus comes gonadotropin releasing hormone. This goes and affects your anterior pituitary, right? And the anterior pituitary produces gonadotropins, which are of two types. One is FSH and second is LH. Please tell me these points are clear. Yes. Now, both FSH and LH are going to act on different structures. FSH is going to act on Sertoli cells, while LH is going to act on IC cells, that is interstitial cells or Leydig cells. Now guys, because luteinizing hormone is going to act on interstitial cells, we call luteinizing hormone as interstitial cell stimulating hormone, that is ICSH. So LH is also in males, in case of males it is known as ICSH. Please tell me these points are clear. Right? And then what happens to the Sertoli cells? Sertoli cells are going to cause spermiogenesis in the presence of IC cells which are producing testosterone. Please tell me this hormonal regulation is complete. And also I told you the Sertoli cells are known to produce inhibin, guys. And as the name indicates, inhibin is going to inhibit the production, inhibin is going to have negative feedback on hypothalamus and anterior pituitary. Please tell me these points are super clear. Okay, Shalini, I'll do that. Yes, clear? So guys, this is the chart of sperm production given in your NCERT. Let's have a close look. So at puberty, what happens? Spermatogonia, it undergoes mitosis to form and then differentiation to form primary spermatocytes. And primary will undergo first meiosis to form secondary spermatocytes. This will undergo second meiosis to form spermatids, and this will undergo differentiation to form spermatozoa. And we call this differentiation as spermiogenesis. Yes or no? Right, guys? And then, so we have studied this already, right? You must know the ploidy, and then you can easily find out what will be the number of chromosomes in each. So spermatogonia, 2n, and then primary spermatocytes will also have the same Primary, they will undergo meiosis to form. Then secondary spermatocytes are haploid. So they will have how many? 23 chromosomes, right? And then the secondary will have spermatids, which are again haploid, that is 23 chromosomes. Please tell me these points are clear. And last not the least, when these sperms are produced in the seminiferous tubules, after getting formed, guys, it is very important to see the sperm's head is embedded in the Sertoli cells to draw nourishment. To draw nourishment. Is that clear? And the release of sperms, once there is once the sperms have drawn nourishment, they are going to be released from the seminiferous tubule and going to move ahead in the passage till they reach the till they reach the epididymis. Now this is known as spermiation. Very important. What is spermiogenesis? Conversion of spermatid to sperms. What is spermiation? Release of sperms from the seminiferous tubule, from the head into the lumen, and through lumen into the straight ducts, into the vasa retrotestes, then epididymis, right? And vas deferens. So epididymis to vas to epididymis on, till it reaches the epididymis, where they are going to be stored temporarily. Now that is called spermiation. Are we clear? Please tell me these points are clear to all of you. And now moving on to the structure of sperm, guys. The structure of sperm, see here, the structure of sperm consists of 4 parts. One is head, then we have neck, then we have middle piece, and last is the tail. Is that clear? Last is the tail. So till here, guys, we have head, we have a small neck, rest of it is all middle piece, and then the entire structure is called a tail. So what are the components present in head? See, plasma membrane will be present all throughout. The main structures present in head are acrosome and nucleus. Then in neck, guys, we have this structure known as proximal centriole. We have proximal centriole, right? In middle piece, guys, we have this structure which is the spiral mitochondria. We have the spiral mitochondria, and mitochondria is the energy source for swimming. And then also, guys, remember in the middle piece we also have one other structure not mentioned here, but this is the distal centriole. So we have 2 centrioles, proximal in the neck region and distal centriole here. And from the distal centriole arises this structure, which is known as axial filament. This is all axial filament, which is responsible for the movement basically. And now axial filament will be not surrounded by the membrane here. So see, this is all plasma membrane, or you can call it tail sheath. The plasma membrane in the tail region is tail sheath. This is the tail sheath. Do you see that there is some part of the axial filament which is not surrounded by tail sheath, and this part is known as end piece, guys. This is known as the end piece. Is that clear? Where the tail sheath is absent, that part is called end piece. Now let's understand the functions. So acrosome, guys, first of all, acrosome. So what does this acrosome do? Acrosome is basically made up of Golgi bodies. So it secretes the hydrolytic enzymes, and these hydrolytic enzymes are going to help in penetration of sperm into the ovum. So these enzymes are known as sperm lysins. And what does these sperm lysins do? These sperm lysins help in penetration, for penetration of sperm into the ovum during fertilization. During fertilization. Please tell me these points are clear. Sorry, during fertilization. Are we clear? Yes, please. right? Then guys, we have nucleus, and what does nucleus does? It obviously contains the genetic material. How many chromosomes will be present in here? It will contain 23 chromosomes because it is haploid. And we have just seen why haploid, because it is formed as the result of meiosis. Then proximal centriole. This proximal centriole is going to help us during cleavage actually, because ovum does not have its own centriole. So when the ovum has changed into zygote, so this proximal centriole will help in the division of that zygote during cleavage of zygote. That division of zygote is known as cleavage, and that's where proximal centriole functions. Distal centriole will give rise to, this gives rise to axial filament, and axial filament is responsible for the movement or the motility of sperm. And we already know what is the function of mitochondria. And then the tail, this whole tail will show flagellar movement for the swimming of movement of sperm. Please tell me all these points are clear to all. Yes guys, please tell me these points are clear to everyone. Yes guys, quickly, please tell me these points are clear to everyone. Is that clear? Perfect, perfect. Now, moving on to the, so we have already discussed this. Moving on to, okay, now there are some facts about sperms that you must know. So we have already seen there are 4 parts, which are head, neck, middle piece and tail. It is all surrounded by plasma membrane, haploid nucleus. So how many chromosomes? 23. So there is a cap-like structure present known as acrosome. What are the functions? Enzymes which help in fertilization, that is the penetration. We will see there are certain membranes around the oocyte which need to be digested for the sperm to enter. So we know mitochondria, which is going to provide energy. Is that clear? Then how many sperms are produced, or sorry, not produced, present? How many sperms are present per ejaculation? See, one ejaculation contains around 200 to 300 million sperms. Now it is very important guys that out of these 200, 60% must have normal shape and size. And out of these 60%, the 60% which should have normal shape and size, out of these 60%, 40% should show vigorous motility. So total should be 200 to 300 million. Out of 200 to 300, 60 should have good structure, and out of those 60% which are good in structure and size, out of that 60%, 40% should have normal and vigorous motility so that it can reach the ovum or oocyte at the right time. Now remember, what is the viability of sperms? Viability of sperms. How do we decide whether the motility is right or not? So basically, after the sperms are released, guys, viability of sperms means till they retain their fertilizing capacity. Beyond this time period, it will be useless, right? So viability is 24 hours after ejaculation. Before ejaculation, it is for longer time, but after the sperms have been ejaculated, released out of the body of the male, there are only 24 hours within which it has to strike the oocyte. Is that clear? So that is the viability. So it should have sufficient motility that it can reach to the oocyte within 24 hours. Clear to all? Is that clear? Now see, sperm along with the secretions, that will be called semen. So it is sperms plus secretions. Now these secretions are coming from 2, seminal vesicle and prostate gland, these secretions. So semen does not include the lubricant. Is that clear? So the function is maintained by testicular hormone, that is a testosterone. Are we sorted? Are we clear till here? Please tell me guys. Are all these points clear to everyone? So guys, with this we have completed the male reproductive structure and the spermatogenesis. Please tell me these points are clear to all. Please tell me these points are clear to all. Is that clear? Red is not very clear. Okay, I'll take notice of that. All right, guys. Now, all right, noted. All right, guys. Now moving on to the female reproductive system. Now, just like we have studied the male architecture first before we studied the gametogenesis, we are going to talk about the female reproductive system. What does this consist of? This consists of a pair of ovaries, then a pair of fallopian tubes, then uterus, vagina, right, and the external genitalia. Now see, uterus also known as womb, also known as hystera, also known as oviducts. So how many are they? So these are paired, this is one. Vagina is also singular, and then the external genitalia, which is called vulva. We'll be discussing all these structures before we move on to the actual functional aspect of these structures. All right, guys. Now, moving on to the first. So this is how the female reproductive system looks like. This is how it looks like. So these are the ovaries, paired structures. Now, unlike testes, which are in extra-abdominal, ovaries are very much intra-abdominal. So most of these regions, most of the structure except for external genitalia, this is completely intra-abdominal. It is intra-abdominal. Where is it found? It is found in the pelvic region, that is the hip region. Is that clear? So do you see these tubes called fallopian tubes 1 and 2? We see this uterus and we see this vagina. Now we'll be discussing each part one by one. We'll be studying each part one by one, starting with ovaries, guys. So what are the functions? So first of all, how many are they? I've told you they are paired, they are oval, they are pinkish, right? And what is the function? What are the functions? So there are two functions of ovaries, just like in case of testes. One is ova formation, ova production, or we call it oogenesis. That is one. And second is formation of ovarian hormones. That means this is also endocrine in nature. Endocrine in nature. Now, which are the hormones produced by ovary? So one is estrogen, and the second is progesterone. So testes was secreting one male hormone. These are secreting two different hormones, estrogen and progesterone. As we go into the details of ovary and see the structure of the follicles present, we will see which particular cell is responsible for estrogen production and which one is going to release the progesterone. Are we clear? Please tell me these points are clear to all. Yes? All right, guys. Now, moving on to the details here. So, first of all, see here. See this. These are testes. These are ovaries, right? These are ovaries. Now, ovaries are, and this being the uterus. This is the uterus. So, this is how it looks. This is the uterus, right? Now, do you see there are some cord there? This is that fibrous structure which is connecting ovary to the uterus. It is connected to the uterus. And guys, this part is known as ovarian ligament. This part is known as ovarian ligament. Is that clear? This part is known as ovarian ligament. And then these are the fallopian tubes. And so these are the fallopian tubes, right? But this is the ovarian ligament which connects it to the wall of the, ovarian ligament connects ovary to the uterine wall. ovary to uterine wall. And then ovary is also connected to the rest of the abdominal cavity. So how is it connected to the abdominal cavity? It is connected to the abdominal cavity through mesovarium. So this whole yellow structure that you see in here, this whole structure which is connecting it to the body wall, this is called mesovarium. Please tell me, are these points clear? This is called mesovarium. So it is a double layered fold of peritoneum connecting ovary to the wall of abdominal cavity. This is mesovarium. Please tell me all these points are clear. While ovarian ligament particularly connects it to the uterine wall. Is that clear? Ovary to the uterine wall through ovarian ligament. Are we sorted? Right? Now, ovary, like any part of the body which has outer region and inner region, this ovary is also differentiated into two parts. So the outer part, guys, outer part is known as cortex and the inner part is known as medulla. So this is the inner part. Is that clear? Now what does inner part consist of? See here, the inner medulla, it has blood vessels, nerves, right, and connective tissue. It has these structures, while cortex contains the ovarian follicle. Is this clear? Now this whole part, this is all stroma. This is all stroma. So basically the inner part is called, the inner structure is stroma, which is divided into two parts. One is outer cortex and inner medulla, and the outer part, this is all stroma, right? So outer part contains ovarian follicles and the inner part contains blood vessels, nerves and connective tissue. Are we clear? Please tell me. Right? Now also I'll tell you the protective covering. So the protective covering of ovaries, see here. Talking about the protective covering once again. So this is the ovary, right? And we know it is lined by germinal epithelium. This is the germinal epithelium. Just like seminiferous tubule was lined by germinal epithelium, and germinal epithelium had two types of cells, Sertoli cells and sperm mother cells, spermatogonia. Yes. Now similarly, the ovarian wall is also lined by germinal epithelium. Right? Basically, germinal epithelium is cuboidal epithelium, but capable of undergoing meiosis. This is germinal epithelium, right? It is cuboidal. And just below this, we have this fibrous tissue. We have this fibrous tissue, and this fibrous tissue is known as tunica albuginea. Have we read the same fibrous tissue in males also? Yes or no? So guys, this represents the protective covering of ovaries, and present below the germinal epithelium. Please tell me if these points are clear. If there is a doubt, also mention that in the chat. Are we clear? Yes, please. Right, Akshay. Clear to all? Perfect. Perfect. And rest, the whole tissue was known as, stroma, and we have seen that stroma is divided into two parts, cortex and medulla. Yes, we have already seen this. Now guys, moving on to, other than ovaries, now moving on to the structures. See, we have read about the different structures of it. Yeah. So I've told you, like in case of males, we have seen that this part represents the primary sex organ of females. All these structures will represent the secondary sex organs. Yes, we've read about ovaries. In the second part, we are going to talk about how they help in oogenesis. But before that, we are going to talk about the secondary sex organs. That is, first of all, fallopian tubes, right? Now, let's talk about these fallopian tubes here. Fallopian tubes, as we can see, so these are the fallopian tubes, right? These are the fallopian tubes. So are they paired? Yes, they are paired. They are long, they are tubular, and on one side they have ovary. They have ovary on one side and uterus on the other side, right? Now, so can we say it connects ovary to the uterus? Yes or no? So we can say it connects ovary to uterus. Also guys, each fallopian tube, also known as oviduct, because it is going to pass, it is going to become a passage for the ovum. It is going to help in the transport, right? Now, each oviduct has 3 regions. Each oviduct has 3 regions. One is this, then this portion and this portion. Let's understand. So there are 3 parts. One, infundibulum. Then we have this isthmus. Sorry, we have this ampulla. And the third part, which represents the isthmus. So these are the 3 regions of or parts of oviduct. Please tell me these points are clear. There are 3 parts. Can we say infundibulum is a part near the ovary, right? And it is funnel shaped. I'll draw the structure to explain that as well. It is funnel shaped and it has these, can you see these finger like processes that I've drawn here? So it has these finger like processes known as fimbriae. Please tell me these points are clear to all. Yes, please. So these are fimbriae. Yes. So what are they going to do basically? Let me draw the structure of infundibulum here. So guys, this is how it is a funnel shaped structure. And there is this opening known as ostia. And around these ostia, guys, we have these these finger-like processes known as fimbriae. These are fimbriae. Is that clear? These are fimbriae. So basically like this. So these fimbriae are present right over the ovary like this. So whenever the ovary is going to release the oocyte, these fimbriae are going to pick them up. Is that clear? So this is infundibulum, the opening is ostia, and these finger-like processes are called fimbriae. What is the function? They help in picking the oocyte. They pick up the secondary oocyte during ovulation. Are we clear? Is that clear? So they pick up the secondary oocyte during ovulation. Are we clear? Yes, and pass it on to the next part, which is called ampulla. So what is the function of ampulla, guys? Ampulla, number 1, it is the dilated region. What is ampulla? Any region which is dilated, we refer to it as ampulla, right? Dilated region, and hashtag, this is the site of fertilization. So basically, this is the site of fertilization. So see here, so basically the sperms are also going to reach here and the oocyte is also going to be present here, and this is the location where they will fuse. So fusion will take place, site for fertilization. Are we sorted? Yes. And then ahead of ampulla we have this isthmus, which is rather narrow region, narrow, and connects fallopian tube to uterus, right? So it is a part near the uterus, part near the uterus. Is that clear? Now the whole fallopian tube, guys, is lined by, listen very carefully, it is lined by ciliated columnar epithelium. Yes? Have we read about this in our structural organization? Yes or no? Have we read about this in structural organization? Please tell me. Why is cilia present? It is this cilia, guys, which is going to help in movement of ovum or even embryo gradually towards the uterus. Also, your fallopian tubes show peristalsis. They show peristalsis. So there are two movements seen in fallopian tubes. One is peristaltic motion and second is the ciliary motion, which helps in the movement towards the uterus. Please tell me these points are clear to everyone. Yes? Yes, guys, please let me know these points are clear to everyone. And now moving on to the next, that is uterus, also known as womb, also known as hystera, also called hystera. And we know that there is single womb. It is large, it is muscular, highly vascularized. Highly vascularized means lot of blood supply here, because if there is no rich blood supply, the uterine wall will not develop. So this whole structure is the uterus. Till here we have uterus. Do you see in this diagram? This is the uterus. This is the uterus, and your uterus is hollow from inside. It has to be hollow. Can we say it is also dome shaped? Can you see the shape? Dome shaped structure. Yes? Do we see this dome? This is the dome. This is the dome. Yes? This is the dome. And now, guys, we can divide this whole uterus into three regions. We call this part as fundus. This is the body. And this posterior part is called cervix. So, uterus is divided into three regions. Fundus is the dome part, then the main part is called the body, and third part is called the cervix. Please tell me these points are clear. Yes or no? Clear? All right. Now see. Talking about the wall of the uterus. So, uterine wall, see, I told you uterus is hollow from inside. So, this is all the uterine cavity, right? Now, there are three parts already discussed: fundus, the dome shaped, then body, the main part, and this is cervix, the posterior part. This is the posterior part of cervix, which is narrow, right? And in this part is the cervix. Clear? And in this cervix, the canal in the cervical cervix region is called cervical canal. And along with vagina, so this becomes vagina. This is the vaginal part. So vaginal canal and cervical canal together, they are going to make the birth canal. We know this, right? Now talking about the wall. So guys, the wall has three layers. The wall of uterus has three layers. One is perimetrium, we have myometrium, and we have endometrium. Very, very important aspect now. So please read carefully with me. So perimetrium, see here. So this outer part is the perimetrium, right? Then we have this as the endometrium. This whole part, this is the endometrium. Similarly on this side, and the part in between this region, I'm shading as yellow, this is the myometrium. So this is the middle, this is the outer, this is the inner. Is that clear? So this is outer, middle and inner. Endo means inner. Myo doesn't means middle. Myo means muscles, and peri means outside the boundary. So it is made up of external thin membrane and connects uterus to the body wall. So what is the function here? Connects uterus to the body wall. Yes. Then we have myometrium, guys. What type of muscles are present here? Smooth muscles. That means involuntary. Of course, the uterine contractions are not in your control. So hence involuntary. So during menstruation also, the cramps that we feel, or during parturition, the labor contractions that happen, they all occur in the myometrium. So this is responsible for, guys, this is responsible for labor. Labor are the strong contractions during childbirth. So it occurs here. So which is that hormone which induces labor, which induces contractions? That is oxytocin. So actually oxytocin is going to act on these smooth muscles. That means which layer? Myometrium. Are we clear? Right? And then we have endometrium, guys. Endometrium is highly vascularized, highly glandular and tissue system. And also it undergoes cyclic changes every month, shows cyclic changes every month as per the menstrual cycle. So yes, during every cycle it develops and it is also shed off. So cyclic changes are shown by this particular layer. Is that clear? Please tell me. It is vascularized. This vascularization will help in the development of more development of the layer, and glandular because it can secrete hormone. Yes, it can secretes estrogen during the luteal phase. We will see it during the menstrual cycle. Please tell me these points are clear to all. Right, guys? And then we have vagina, and this part is called vagina, which is tubular, and it has some folds which are called, it has folds to increase the surface area. Also it has lactic acid bacteria. It has lactic acid bacteria for producing lactic acid, and lactic acid is going to protect us against many infections. So this lab produces lactic acid, right? And this lactic acid provides the primary immune response. Sorry, not primary immune response. This provides the morphological and anatomical responses, non-specific immunity. This is a non-specific immunity. Please tell me these points are clear to everyone. And this is so, this lactic acid is so acidic that the pH in our vagina is around 3 to 4, which again is going to prevent many infections from happening. Are we clear? Please tell me. Right? And together with the cervical canal, I've told you it forms the birth canal. Is this clear? And then talking about the female external genitalia, and because it is, why do we call it external? Because this part is of course extra abdominal. This is present outside the body. Outside the body. Right? Now, see very carefully. So there is this cavity called vestibule. Wait, I'll draw it here. Now see, so we have this structure. Here we have our structure called anus. This is the posterior region. This is anus. And guys, here we have this whole cavity. This cavity is known as vestibule. This is called vestibule. Vestibule means cavity, the depression. Clear? And in this vestibule, we have two openings. One is the vaginal opening or vaginal orifice. And on the upper side, we have urethral orifice. That is opening of urethra. Now, unlike in males, in females, the excretory and reproductive system are totally separated, right? Is that clear? Now see, here we have some hairy and highly fatty tissue. This area is known as mons pubis or known as mons veneris. Is it clear? These are the parts. It is characterized by pubic hair and subcutaneous fatty tissue. Subcutaneous means below the skin. Subcutaneous fatty tissue. Is this clear? Right? Do we see these openings here? Now guys, surrounding the urethral orifice, we have this fold. This fold of skin is known as labia minora, also known as lesser lips. Is it clear? This is known as labia minora or lesser lips, while on the outer side guys, here we have labia majora or the greater lips, because they are bigger in size. Is this clear? Now one very important structure I want to mention. So do you see these structures? This is labia minora, right? This is all labia minora. Now here in the upper region, here, where labia minora fuse, the two fleshy folds of labia minora fuse, this is clitoris. Now just like in males, glans penis, that is the tip of penis, is called the main erectile part. Similarly in females, this is the female erectile part. With lots of free nerve endings. Is it clear? This is clitoris. And where is clitoris present? See here, where is clitoris present? It is present at the upper junction. Present at upper junction of labia minora. Clear to everyone? Please tell me. Right? Upper junction of labia minora. That is clitoris. This is a female erectile part. Right, everyone? Now, let's talk a little bit more about majora and minora. So, majora, they are outer, thicker, hairy, and with sweat glands. These are the features of labia majora. While talking about minora, these are inner, these are thinner, these are non-hairy, they are moist but no sweat glands. Are we clear? Please tell me these points are clear. So, this is vestibule. Now, surrounding the vaginal orifice, now see one more thing. Vaginal orifice, that is the vaginal opening, it is covered by a membrane known as hymen. So there is this membrane called hymen present in here. This is where hymen is present. Is it clear? And this hymen is going to break. Number one, it is going to rupture, number one, during the first menstruation. Also, it will break during the coitus, first coitus, but it can also break during the certain physical activities like cycling, like when there is a sudden fall or a jolt, or during horse riding, right? So there are many ways in which this hymen can break. It breaks during first coitus, right? Then sudden fall, gymnast, horse riding, etc. Cycling, etc. So that is why it is not rightly known as a virginity knot, virgin knot. So we cannot call it the sign of virginity. So not a true indicator of virginity. Is this clear? Not a true indicator of virginity. Now guys, around these two openings, we do see some other structures. For example, around the urethral opening, we have these glands which are called Skene's glands or paraurethral glands. Around the urethra, we'll call them paraurethral. Is that clear? Paraurethral. So what is the function of Skene's glands? They are going to produce slightly alkaline fluid. They secrete alkaline fluid to neutralize the acidity in the vaginal area. Why? Because sperms cannot survive the acidic conditions. So before the entry of sperms, the Skene's glands are going to produce the alkaline fluid to neutralize that acidity. Is that clear? Now, and they are smaller, and these are smaller, these are 6 to 8 in number, right? Smaller. So these are the characteristics. So we also call them lesser vestibular. Okay. Now surrounding these, surrounding this vaginal orifice, we have large sized two glands. These two glands are known as Bartholin's glands. In the name of scientist, Bartholin's glands. How many are they? 2 in number. What do they do? Lubricate. They help in lubrication. Can you tell me in the chart which gland in males serve the same purpose? Yes guys, which gland in males serve the same purpose? Yes please. Any class after the session? Toyota, I have plus class after the session. Copper's gland, right Lakshya. So we have Copper's gland, also known as bulbourethral glands in males, serving the same purpose of lubrication. Is this clear? So this is how guys, this is how our external genitalia looks like, which is known as vulva. This is vulva, the external genitalia of females. And also in this vulva, we have seen two main glands. One is the paraurethral gland, and second we have seen the Bartholin's glands. Are we sorted? So we have seen the external genitalia, and along with the external genitalia we have also seen the accessory glands in females. Please tell me all these points are clear to you. Yes guys, please let me know. Are we sorted? Are we clear with the structure? Right? Okay. So now guys, moving on to, so glands already discussed, see. So we have Bartholin's glands, we have Bartholin's glands also. Because they are bigger, they are called greater vestibular glands, and what do they do? Lubricate. Then we have Skene's gland also, because they are smaller, so we call them lesser vestibular, and because they are present around the urethra, we also call them paraurethral glands. And what do? They produce slightly alkaline fluid. And the third type of glands which are present in females is the mammary glands guys. These are mammary glands. Hashtag unique feature only of mammals. Unique to mammals. They're highly glandular because they secrete milk. Is that clear? Right? Now, talking about the mammary glands here. So mammary glands, how do they look like? First, let's see. So guys, this is the back side of the dorsal side of the body. These are the ribs, and in between ribs we will have, in between the ribs we have these muscles, intercostal muscles, right? Then ahead of it lies the pectoralis major, that is the pectoralis major muscles. So these are ribs. In between we have intercostal muscles, ICM, yes, and then we have these pectoral muscles. Now let me show you the structure of mammary gland here. Now see, in a mammary gland, in one mammary gland, there are around 15 to 20 such structures known as mammary lobes. How many mammary lobes? 15 to 20 in number in each mammary gland. Is that clear? Now, within these mammary lobes, guys, we have these sac-like structures called alveoli. What are these alveoli going to do? These are sac-like structures and they are lined by some cuboidal epithelium which is going to secrete milk. This secretes milk. Is this clear? So this is alveoli. Ahead of alveoli lies these tubules. Is that clear? And then we further have these tubules. So there will be so many tubules coming out of each lobe like this. Are you getting it? Please tell me these points are clear. Right? So guys, these red, so we have these red tubules called the mammary tubules. And so many mammary tubules will open up in a common mammary duct. Now these mammary ducts, guys, they will open in this dilated portion. What is dilation known as? Ampulla, mammary ampulla. Yes or no? And from the mammary ampulla arises, so we'll have so many ampulla, and from these ampulla arises these ducts called lactiferous ducts. So they are around 14 to 16 in number. These lactiferous ducts will together open in one area called nipple. So nipple is the multiporous area. So it has openings of these lactiferous tubules. Please tell me. And now the question that comes up usually is what is the correct sequence here? So first of all, you must know what are the structures. So we have mammary lobes, 15 to 20. In every mammary lobe, guys, we have alveoli which secretes milk. From alveoli arises the mammary tubules which are going to carry this milk ahead, and from mammary tubules becomes mammary ducts. Then we have ampulla which stores milk. So this produces or secretes milk, while this stores milk, and this releases milk. So when the milk has to be released, ampulla is going to contract where the milk is stored and it will be released out through the lactiferous ducts. Please tell me these points are clear. And in between, guys, there is this space which is all filled with fatty tissue. So there has to be fatty tissue here, which gives it a proper contouring as well. Is it clear? Now, mammary glands are under the effect of 4 different hormones. Now, see, number 1 is estrogen. Number 2 is going to be progesterone. Number 3 is oxytocin. And number 4 is prolactin. What does each one of them do? Function of estrogen, progesterone, oxytocin and prolactin. What are the functions of each? See, first of all, this fatty tissue, the deposition of fatty tissue is because of estrogen. This is because of, I'll write it down, because of estrogen. Mammary lobes, they develop because of progesterone, due to progesterone. Alveoli produce milk because of prolactin, due to prolactin. The prolactin produces milk. So where is it going to act? Alveoli. And then milk is released due to oxytocin. And more the milk is released, more prolactin will be produced. So this is going to have positive mechanism here. positive feedback mechanism. Please tell me all these points are super clear to everyone. Yes, guys. Are we clear? Yes, are we clear? Anyone with any doubt? Clear? Crystal clear? Perfect. So this is the structure and this is already a PYQ. Make sure that you know the sequence. And now guys, coming on to the process of oogenesis. See here. Process of oogenesis. So we know this oogenesis, see, unlike spermatogenesis, so when, where, and why, and how, right? These are the questions we are going to answer. So when does oogenesis begin? It begins it begins before birth. Unlike spermatogenesis, which occurs or which starts after birth, that is during puberty, this begins before birth, then stops at birth or pauses at birth. Let's say it says pauses at birth and then restarts at puberty. Restarts at puberty. And unlike sperm production, which will go on throughout the lifetime, this doesn't. It will stop permanently. Stops permanently at a certain age, which is called, and we call it menopause, around 45 to 50 years of age. That is menopause. So do you see, it is not occurring continuously. Hence, oogenesis is, guys, a discontinuous process. Can we say it? It is a discontinuous process. Yes or no? Right? Then where does it occur? It occurs in the ovary and particularly in the ovarian cortex, in the follicles, right? Why? Again, because of the hormones. There is hormonal regulation. We'll talk about the hormonal regulation in females as well. What happens during puberty that it restarts? We'll talk about it all. How? Now, for that, we need a separate slide. So see here, guys. First of all, this is your ovary. This is your ovary, right? And we know that ovary is lined by this germinal epithelium, which we have already discussed. And below the germinal epithelium, we have tunica albuginea. Yes. And we also know that there is this whole stroma present. I'm not drawing the medulla here just to save space for the drawing of the follicles here. Is that clear? But of course, we have medullary region in the center. So this is all stroma, and of course, stroma contains certain cells. Now, starting with that why it started. Why it started? Because, see, first of all, it started before the birth, that is during the fetal stage. Now, what happens? This germinal epithelial cells, which represent the ovum mother cells, right? These ovum mother cells are going to come and lie in the stroma. Yes. Now, what happens to these ovum mother cells? Let's first see it with the help of a flow chart. See here. So just like a sperm mother cell, we have ovum mother cell, also known as oogonia. Now, just like it happened in case of sperm mother cells, there will be two types of cells, out of which one will continue to divide, other will go into a stage of growth, right? And so on, right? We call it type A and type B. This is type B, ovum mother cells, ovum mother cells. So these are B, B, and so on, right? And all of them, they are 2n. So they have 46 chromosomes. They all are 2n, 2n, 2n. And we know that they are all undergoing mitosis. So this phase is called the multiplicative phase, where they are just increasing their number. Is this clear? So these ova mother cells are going to undergo mitosis and increase their number, right? Now what will happen? These type B cells, they will show growth. They will show growth and enter the growth phase. So they will just grow in size and enter the growth phase. So like this, they will just grow. And after growth, guys, see, these are ova mother cells, or we call them oogonia, right? After growth, they become primary oocyte. There's no change in the ploidy, which still remains 4n, that is 46. Are we clear? Yes. Each is going to enter the growth phase, but I am going to continue with one oogonia and its future destiny. Is that clear? So this is a primary oocyte. Now after this primary oocyte is formed, it is going to undergo meiosis 1, and this is your maturation phase. So we understand that during maturation phase, it is the meiosis that takes place. Yes or no? It is the meiosis that takes place. So when meiosis 1 takes place, now listen very carefully, guys. Meiosis 1. During sperm production, what happened? It gave rise to 2 equal cells. Yes or no? But here, in case of females, when meiosis 1 occurs, there is equal distribution of the genetic material. So this gets half, this gets half. But when it was time for the division of cytoplasm, that is cytokinesis had to take place, now there was partiality. One of them got very less cytoplasm, while the other got most of the cytoplasm. So in males, there were 2 similar secondary spermatocytes formed, but in case of females, the larger one, which gets a maximum cytoplasm, is known as secondary oocyte, right? This is the secondary oocyte, while the other, which remains small, this is called the first polar body. Please tell me these points are clear. So in meiosis 1, there is equal karyokinesis, equal nuclear division, but unequal cytokinesis. That was not the case in spermatogenesis. Do you see the difference here? Yes or no? Now guys, time for meiosis 2. Now this polar body, because it has very less cytoplasm, this will undergo degeneration. This undergoes degeneration. It degenerates. Sooner or later, it is going to degenerate. It might undergo meiosis 2 and then degenerate, like it is the case with humans, but NCERT doesn't mention. NCERT simply says that it degenerates. So it degenerates. Is that clear? Because of less cytoplasm. So do we realize that cytoplasm is as important as nucleus, because most of the metabolic processes are occurring in cytoplasm. So without cytoplasm, we cannot survive, but without nucleus, some of the cells do survive, right? Anyways, so this is the secondary oocyte, which is going to undergo meiosis 2, right? Now, when it divided, again, there was the same pattern observed. Equal karyokinesis and unequal cytokinesis. So again, it gave rise to one big cell, which will be known as ovum, and a smaller cell, which will be known as second polar body, because it is formed during the second meiosis. And again, it will have a same destiny like the first polar body. This will also degenerate. Please tell me these points are clear. Now we said that all these processes are discontinuous. They don't occur in the same flow. It will not occur in one flow. They will occur discontinuously. So one process will happen, then it will take a pause, then it will restart again, and so on. Now, what are the processes which occur before birth and then later on after puberty? Now see, we talk about meiosis 1. We know that in meiosis 1, the prophase We know about prophase 1. Yes. We know about metaphase 1, anaphase 1, telophase 1. Yes. Now see, in prophase 1, guys, in prophase 1, there is a phase called diplotin. So, see carefully, the process till diplotin occurs before birth. This occurs before birth, right? It stops at diplotin. And then after diplotin, rest of the stages, that is dikinesis, then metaphase 1, anaphase 1, telophase 1, this will all occur after puberty. This is after birth, that is at puberty. Do we understand it? So when we are born, we have around, so see, till meiosis 1 is not completed, we are going to call it primary oocyte itself, but primary oocyte which is arrested in diplotin stage, it is arrested at diplotin stage. So this is at birth. So we are born with primary oocytes arrested in diplotin stage, and how many? Around 1 million such primary oocytes at the time of birth in each ovary. 1 million. Do we see that? And then after puberty, at puberty, so in between, guys, there is a gap of 10-11 years. So during that gap of 10-11 years, these 1 million primary oocytes are going to degenerate. Is that clear? Is that clear? So by the time we reach our puberty, we are only left with 60,000 to 80,000 follicles or oocytes. Is that clear? Now, primary oocytes. Now after that, there will be secondary oocyte. It will form at puberty. One of them is going to form secondary oocyte and one polar body, and then meiosis 2 and so on. Now see, in meiosis 2 also, things will not happen in one flow. So we have prophase 2, we have metaphase 2, we have anaphase 2, and we know there is telophase 2 followed by cytokinesis. Yes? Now see again, once this primary oocyte has started its journey forming secondary oocyte, it enters the meiosis 2. See, it will again stop at metaphase 2 stage. This is the second pause it is going to take. Now, this will pause. So by the way, at the time of by the time secondary oocyte and first polar body is released, it will be released out of ovary. So ovulation is going to take place here. And now listen, this is going to have a second pause. This will be second pause. Now if we want that secondary oocyte should complete the life cycle and become ovum, for that we need a sperm entry here. So it is the sperm entry which is important for the meiosis 2 to complete and form ovum. It means if the sperm doesn't enter, it never becomes an ovum. It only remains as secondary oocyte which has entered the meiosis 2 but not completed it. Please tell me these points are clear to everyone. Hi Srija. Srija, you can definitely subscribe to the English medium Phoenix batch 2.0, right? But we are going to start with the Phoenix batch, but the classes have begun. Some students who have already enrolled for Phoenix batch, we have started taking their classes, but the proper launch of the batch will take place after Re-NEET. Is that clear? You can enroll and attend the classes somehow. Please tell me these points. Are these points clear to all? So you can enroll for Phoenix 2.0 anytime. Clear? It is just a covering, Akshay. No such function as such. All right. All right. So this was about ovulation. Now, so I was telling you about ovum mother cells. So they will undergo multiplication, and after multiplication, see, they are going to get bigger. And once it is bigger, we call it primary oocyte. Right? Now see, there are some cells present in the stroma. These cells will also surround the primary oocyte. right? Like this. So these have been derived from the stroma, and because it forms a flower-like structure, these are called follicular cells. And the cell in the center is called the primary oocyte. Is it clear? Now gradually this primary oocyte is going to remain as such. So this is primary oocyte, but the follicular cells surrounding, they will increase in number. So this is follicular cells, they become multi-layered, and now it will form a second follicle. Now see, this one is called the primary follicle, and this is called the secondary follicle. Please tell me, right? So in primary follicle, we have primary oocyte in the center and one layer of follicular cells, while in secondary follicle, we have primary oocyte as such, and the follicular cells have multiplied and become multi-layered. Yes. Now, after this, guys, what will happen? This primary oocyte is going to undergo meiosis 1. It undergoes meiosis 1, and what will it form? It will form secondary oocyte along with the first polar body. Yes, first polar body. Are we clear? Now, about the follicular cells, these are the follicular cells. These follicular cells are going to, see, these are all follicular cells. These are all follicular cells, right? These follicular cells are going to form theca. Like this. So there will be two coverings around it. So we call it theca externa, and this is theca interna. Also, the secondary oocyte has now released a covering around it. So this covering around the secondary oocyte is known as zona pellucida. Very, very important layer. Is this clear? And guys, this represents the tertiary follicle. Is this clear? Now, after this guys, the secondary oocyte will remain as such. This is the first polar body surrounded by the zona pellucida, which has been secreted by secondary oocyte. This secretes zona pellucida, and now we have these cells. These follicular cells are now known as granulosa cells here. Is that clear? It has these granulosa cells. Now granulosa cells are going to secrete, and they are going to secrete a fluid which will form a cavity. So this will be all granulosa cells, and they have formed a cavity around this. So these are all follicular cells. Is it clear? And then we do have this theca externa and interna. This is theca externa, theca interna. The red ones are the granulosa cells. Now, very important part here is this cavity filled with fluid, which is called antrum. This antrum is fluid filled. This is fluid filled. Are we clear? Right? Now guys, rest of the structures are just the same. The zona pellucida, the secondary oocyte, the first polar body, they remain the same. Now this follicle is called the Graafian follicle. Right? Also, because it has antrum, we also call it antral follicle. And this is the mature one, so we call it a mature follicle. Is that clear? So which one is the antral follicle? The Graafian follicle. Now there is fluid present in the antrum. Who has secreted it? It is secreted by the granulosa cells. Now, what are the functions of granulosa cells here? So these granulosa cells secrete a lot of estrogen and also some progesterone. So you will see as the size of the follicle goes on increasing and the granulosa cells go on increasing, the amount of estrogen goes on increasing in our blood. If this estrogen was high here, estrogen will be very high by the time graphene follicle is formed, and it is usually formed around 11th to 12th day of menstrual cycle. We'll come to menstrual cycle as well. Now, so when the estrogen level is very high, see here, the estrogen level will be very high around 11th and 12th day. When this level remains very high for around 24 to 48 hours, then within another next 2 days, guys, this follicle is going to rupture. So, graphene follicle ruptures. What will happen when it ruptures? Secondary oocyte will come out along with the first polar body, along with the zona pellucida which has been secreted, right? And some of the granulosa cells like this. These are the granulosa cells. But now, because they form an outer structure, we call them as corona radiata. These corona radiata are derived from granulosa cells. It does have this blue colored layer called zona pellucida, and we understand this is secondary oocyte and first polar body. So this graphene follicle ruptures and they all come out of the ovarian perimeter. Now this process is known as ovulation. Please tell me these points are clear. This is called ovulation. Yes, guys, are we clear? Yes, please. Phoenix batch, we complete the syllabus, Rija, by December. I'll try to complete it as soon as possible, even before December probably, but December is our last limit. Is this clear? I doubt that we'll be able to complete a reproductive health because at 7:15 I have my plus class. Don't worry, we'll be able to complete it in the next class, because tomorrow I don't have any plus class. So we'll start at 5 o'clock tomorrow, and we have to complete 3 chapters then. If not reproductive health today, then tomorrow, along with evolution and health and diseases. Right? Clear? Okay. Now, in case I think there are high chances, because I don't want to rush through. We can take extra class, we can take another class, but we have to completely revise the topic in detail. Yes, so don't mind that there will be another session. So see, wait, so there is a possibility, guys, there is a possibility that there is a little bit change in the plan. So that's what I planned. Yes, I think we are going to change this. Yeah, so this is part 1. I'm going to complete this. Then reproductive health, evolution, and probably microbes will be discussed here. Microbes I'll discuss here. And here I'm going to take health and diseases. So this might be the plan now. There might be change in the plan because there is upper limit to the session. So we'll have to stop after few minutes. All right. Is that clear? Noted? So this was, okay. Are we clear? So guys, when it ruptures, the remaining part of graphene follicle forms this yellow colored body, and we call this body as corpus luteum. Corpus means body, luteum is yellow. So we also call it yellow body. Now remember, once the graphene follicle ruptures, the level of estrogen is going to fall, and corpus luteum, because it produces lots of progesterone as compared to estrogen, the level of progesterone is going to increase in blood, right? This corpus luteum has an age span of 14 days if no fertilization occurs. So after 14 days, it is going to degenerate to form the white colored body, which is called white body or corpus albicans. Please tell me these points are clear to all, right? So guys, let's have a quick reading here of certain facts. Let's have a quick reading here. Corpus albicans, no, corpus albicans doesn't have a function. It is the last stage of corpus luteum. So it is going towards degeneration. It will degenerate and will be reabsorbed in the Hanji. Is that clear? So this is oogenesis we are talking, guys. It starts during embryonic stage around 25th week of the fetal stage, and germinal epithelium is going to produce millions of gamete forming cells called gamete mother cells or ova mother cells or oogonia. No oogonia is formed and added afterwards. These lines are very important. Now oogonia enters the meiosis 1 and produces and proceeds to dikinesis and gets suspended at this stage. This is diploteny stage, not dikinesis. Is that clear? And now we call this as primary oocytes. We still call them primary oocytes. Till the meiosis 2 will be completed, we will still call them primary oocytes, and they are surrounded by layers of granulosa cells. So at puberty, only 600 to 8,000 primary oocytes are left in each ovary. Rest of them are degenerated. So at birth, there were 1 million in each ovary. And by the time of puberty, we only have 60 to 80,000. Is that clear? Then get surrounded by more number of granulosa cells and they become secondary follicle and new theca. Is that clear? Then tertiary follicles characterized by, they also have some antrum and they are organized into theca externa and interna. Please tell me these points are clear to all, right? And then it will give rise to two unequal haploid cells. The large cell will be known as secondary oocyte and the tiny cell is called the first polar body. Please tell me these points are all clear. Yes, guys. Is it clear? So see, we can have a little change here. We can have a little change. We can introduce another term, that is, this is of course the tertiary follicle. This is called the secondary follicle. Okay, now this remains the same. This is called primary follicle, and in between, sorry, here we have this one small stage which will be known as primordial follicles. Let's form primordial follicles here. Now primordial follicles will be like this. Okay, so these are primordial follicles that I've drawn. Is this clear? Now see, so this is primordial follicle, and the only difference is in primary follicle we have multiple layers of follicular cells. Clear? And in secondary follicle, basically the follicular cells, they will start differentiating into two types of cells. We'll have theca cells and these theca cells, these are theca cells, and theca cells will become theca in the tertiary stage. Is it clear? So just introduce this change and that's it. Are we clear? All right, guys. So we have completed oogenesis. I've told you all about oogenesis. This is a clear differentiation between spermatogenesis and oogenesis, the number of chromosomes per cell. We have discussed it all. And now coming on to coming on to menstrual cycle. Read it very carefully. Now this is the reproductive cycle, guys, of female primates only, not of all the mammal, only primate mammals, which includes monkey, apes, human beings, right? And menstruation, this is a repeated cycle. It is repeated at an average interval of about 28 to 29 days. While in other mammals, guys, in other mammals, we have estrus cycle. We have estrus cycle. Now those organisms which have estrus cycle, they are not fertile all throughout the year. They are seasonal breeders, while we are continuous breeders. Is this clear? Those with the menstrual cycle, they are continuous breeders. So humans can get pregnant all throughout the year. Is that clear? Now this beginning of the first menstruation, guys, first menstruation is known as menarche. That happens during the age of 10 to 11 years of age. While the major events which occur during the menstrual cycle, one is the menstrual phase, also known as the bleeding phase, follicular phase, ovulatory phase, and luteal phase, also known as the secretory phase. Is this clear? So all these events are going to be divided within 28 to 29 days. Clear? So this is the graph given in here, and you should be able to label the things very properly. I'll explain the graph as well, but you should be able to label. So this line represents estrogen and this represents progesterone, FSH, LH, they have all been put together here. Now this is a different diagram from NCERT. I have taken it particularly because there are many questions given in your practice books where they've asked you to label the hormones. So make sure that you're able to compare and label them properly. Is this clear? Now, talking about what happens during each phase, let's have a quick look. So guys, menstrual phase is the first phase. Now during this phase, basically menstrual flow occurs. So there is bleeding. It lasts for 3 to 4 days and endometrial lining breaks down. The mucus and the blood comes out along with the unfertilized secondary oocyte. It occurs only when ovum released, but no fertilization occurs, right? So lack of menstrual cycle, very important line, is the indication of pregnancy. This is already a PYQ line. Is that clear? So these are very, very important lines. Now coming on to the second phase of menstrual cycle, it is when the graphene follicle, it is during this phase that primary follicle becomes the graphene follicle. There is regeneration and proliferation of the endometrium. Now LH and FSH level go on increasing gradually. Level of estrogen increases maximum during the 11th, 12th day and this estrogen comes from the follicle. We know from the granulosa cells, lasts for 5 to 13 days. 5th to 13th day this will last, right? Then ovulatory phase, this is the shortest phase, by the way, shortest phase, and it is during this phase that both LH and FS peak level. So this occurs during the 14th day. We call it LH surge. What causes LH surge? It is the high level of estrogen. Estrogen very high for 24 to 48 hours causes positive feedback on anterior pituitary, which will release more of LH surge, and this induces the rupture of graphene follicle and causes ovulation. So which hormone is majorly responsible for ovulation? It is the LH surge. Is that clear? Please tell me. And then we have the luteal phase. The remaining part of graphene follicle transforms into corpus luteum, which produces large amount of progesterone and it maintains the uterine endometrium. Now see, remember, endometrium develops due to estrogen. Endometrium develops due to estrogen. Clear? But it is maintained by progesterone. Is that clear? It is maintained by progesterone. Please tell me these points are clear to all. Is it clear? So thickness increases many folds, and this corpus luteum also grows further till the menstrual cycle and pregnancy continued. So in the absence of fertilization, corpus luteum will degenerate if there is no fertilization. Is that clear? And degeneration of corpus luteum means low level of progesterone, and then it will cause the menstrual flow to happen again. So this ceases, menstrual cycle ceases around 50 years of age, and this is called menopause. Please tell me all these points are clear to you. Please tell me, guys, all these points are clear to you all. So this is your NCERT picture for menstrual cycle. Now see, till this day we have, this is your menstrual phase. Then we have, we have this as a we have a follicular phase. Then from here to here, so guys, there's some electricity short. So I will be able to complete the chapter in the next session. I'll complete this, and along with that we will complete our reproductive health. So there's some issue with the board, I guess. So let's continue the chapter in the next session. Make sure that you revise these things, and we'll get back to this chapter. A little part is left, and reproductive health and evolution in the coming session. So thank you so much, and don't forget to, guys, take All India mock tests from happening every twice a week, that is every Wednesday and Sunday. I'll see you tomorrow. Thank you so much.